Transport into and out of the cell. Selectively permeable is a characteristic of a living membrane. The substances that pass through a selectively permeable membrane change with the needs of a cell. For example, the axon membrane of a nerve cell consists of gated channels that open and close to allow specific ions to pass through only when triggered by certain stimulus. Another important vocabulary word you should know is solvent, which is the substance that does the dissolving, like water. Water is known as a universal solvent. Another vocab word is solute, which is the substance that dissolves, like salt and sugar. Hypertonic means having a greater concentration of solute than another solution. So, for example, having more salt than H2O. Hypotonic means having a lower concentration of solute than another solution. And isotonic means two solutions containing equal concentrations of solute. Passive transport is the movement of molecules down a concentration gradient from a region of higher concentration to a region of lower concentration. Passive transport never requires energy, or ATP. It occurs either by diffusion, simple diffusion, or facilitated diffusion, or by osmosis. Simple diffusion is the movement of particles from a higher concentration to a lower concentration. The steeper the gradient, the faster the rate of diffusion. For example, you can think of it as a hill. You don't need energy to go down the hill if it's slanted downwards, and you'll go faster if it's slanted more downwards. Humans obtain oxygen by simple diffusion across moist membranes in air sacs called alveoli in our lungs. Facilitated diffusion relies on special protein membrane channels to assist in transporting specific substances across a membrane. So facilitated diffusion uses proteins. Osmosis is the diffusion of water across a membrane. Water will flow toward the region of higher concentration of solute and lower concentration of solvent. So remember, water diffuses toward the hypertonic areas. Water leaving the cell causes the cell to shrink, and water entering the cell makes the cell swell, and plant cells that swell become turgid. In isotonic solution, water diffuses in and out, but there is no net change in the cell. Active transport is the movement of molecules against a gradient, which requires energy, usually in the form of ATP. For example, you can think of going up the hill. You need energy. You can't go on a sled and expect to go up, but you need energy to go up the hill. Exocytosis is the active release of molecules from a cell. Endocytosis is the process by which cells take in various molecules and particles by forming new vesicles made from the plasma membrane. There are three examples of endocytosis, which is pinocytosis, phagocytosis, and receptor-mediated endocytosis. So pinocytosis is known as cell drinking, which encloses them in a vesicle. Phagocytosis is engulfing of large particles by pseudopods, and the cell membrane wraps around the particles and encloses them, forming a vacuole. And receptor-mediated endocytosis lets the cell take up large quantities of very specific substances. Extracellular substances bind to specific receptors on the cell membrane and are drawn into the cell into vesicles. Another important example of how cells actively transport particles or ions across a membrane against a gradient is the sodium-potassium pump. The sodium-potassium pump is in nerve cells, which carries sodium and potassium across the axonal membrane in opposite directions to return the nerve to its resting state after an impulse has passed. Life functions. A characteristic of all cells is that they carry out certain life processes. They include ingestion, which is the intake of nutrients, digestion, which is enzymatic breakdown, hydrolysis of food so it is small enough to be assimilated by the body, respiration, which is metabolic processes that produce energy, ATP for all the life processes. Transport is distribution of molecules from one part of a cell to another or from one cell to another. Regulation is the ability to maintain internal stability, homeostasis. Synthesis is combining of small molecules of substances into larger, more complex ones. Excretion is the removal of metabolic waste. Egestion is the removal of undigested waste. Reproduction is the ability to generate offspring. Irritability is the ability to respond to stimuli. Locomotion is moving from place to place, animal cells only, and metabolism is sum total of all the life functions. On to the metric system and measuring. So you can look at the chart there. For example, there's kilometers, meters, millimeters, centimeters, etc. And then we often measure with a ruler, so make sure you know how to use that. Another way we measure is using a graduated cylinder, and there is a meniscus, and you want to read the bottom of the meniscus, so it's like a little semicircle, and you want to read the bottom of that. That will be your measurement. Tools and techniques to study cells. There are many tools and techniques to study cells, like microscopes. Compound microscopes study cell structure. When you use the microscope, the images are upside down and backwards. There are also different microscopes, like the phase contrast microscope, which is useful in examining living unstained cells and uses beams of electrons to produce superior resolving power as well as magnification over 100,000. There's a transmission electron microscope, which is useful for studying the interior of cells. However, the tissue is no longer alive under the electron microscope. The scanning electron microscope is useful for studying the surface of cells and also kills the tissue. Other tools for studying cells include the ultracentrifuge, cell fractionation, blenders, freeze fracture, and tissue culture.